cells, tissue, organs, organisms. This all deals with the biophysics and the biology. First of all, what is biophysics? The meaning of the biophysics is a study which applies the chemistry, physics, and even the mathematics in the biological systems. There are three types of biophysics. First one is molecular biophysics, biophysics of the cells, biophysics of complex structure. Now we discuss about these three types of the methods in a brief. First one is molecular biophysics. The meaning of this is structure and the physico-chemical properties of the biological functions. For example, if we take biopolymers, which contains protein and the nucleic acid, the main reason is linked with physics for biological functions. So we use this molecular biophysics. For example, catalytic activity of the protein enzymes. Molecular biophysics is the most developed method as we know, and this we can easily study. One person can easily study the molecules than the cells by this subject called molecular biophysics. And in molecular biophysics also there are many different subunits, that is biochemistry, molecular biophysics, bioorganic, bioinorganic. First we study about the chemical foundations of the biophysics, and then we next chapter we study about the macromolecules of biophysics, and at last we have to study about the molecular physics so that we can understand the sequence of these topics easily. Now previously we talked about the function, structure, everything. Now we talk about the equilibrium. Equilibrium, this is a property of the molecules. We study in the theoretically. What is equilibrium? It is a science of the applications of the laws of physics to biological phenomena. Example, if we take gas physics in respiration, and then diagnostics and in treatment. Now we in this equilibrium we study about the thermodynamics, statistic, and quantum etc. in equilibrium by when we discuss about the equilibrium. This is the theoretically. Now if we take about the experimentally method, in this physical methods are very required, and there is a 4 applications. We can call it as group or method, and the technique to use this. These are the experimental physical method for groups. First group is that macromolecular physics, in which we studies the shape, size, mass. This all is studied by the sedimentation of the ultracentrifugation, X-rays, and the scattering of the lights. And then if we talk about the second group, in this we take the investigation of the molecular structure, like finding the molecular structure and their interaction of the matters of the light by the X-rays to radio frequency. And then we have the spectroscopic technique. In this we use the technique of X-rays, diffractions, and gamma resonance, and electronic and vibration spectra. In this we take the we observe. In this method we observe the light, and then it is luminance, the spectra in the UV rays, and then it is the rays is visible. And then there is a special technique, it is called as another technique, it is called the spectropolarimetry. The meaning is that we investigate or find the natural and the magnetic rotations of the plane of the polarizations of the light. Like in that we have NMR and EPR. The NMR is the nuclear magnetic resonance, and the EPR spectra, and it is very important to learn. And then next we have the third group. In this we include or we conclude only the calorimetric study of how the conversion of the biological macromolecules is converted. And the last but the not least, we study about the group fourth. We study in this proteins and the nucleic acid. In this we can study by our common electron microscope. Easily we can see in laboratory. So all these applications which I told, this is very specific and it is very important. And By the nature, molecular biophysics, which, as I told, it is a structure and functionality of the cells and the tissues. We know that biology, chemistry, and physics is very interlinked. As I told previously, these all are interlinked, and we cannot separable these three subjects because they are very interlinked to each other. And this is the oldest and the traditional as we know till now. And today we study this biophysics for the biological membrane and the bioenergetic process. What is bioenergetic process? It is the transformation of the energy and the storage of the energy in the cell or in the plant also, like a photosynthesis which takes place in the plant. It is a chemistry, like chemistry is used in that. And then if we talk about physics, in that endoscopic we use and x-ray we use, that all the physical method which we use. So we can conclude that chemistry, biology, and physics are interlinked together. Now we talked about the experimental method. Now if we come to the theoretical method, there is a theories sequences, like 4 theories as they're noted in the textbook. First one is a general theory of the dispective non-linear dynamic system, dynamics of irreversible processes. And the second one is that the theory of the excitable media. This includes the oscillatory process also. So what is oscillatory processes? It is a change in the quality or the measurement of the equilibrium. I discussed about the equilibrium theory. So here also the theoretical part is there in equilibrium in a time. Like if we talk about a rope or a clock pendulum, this all the example of the oscillatory processes. And then if we talk, and then the third one is the theory of the bioenergetic process. And the last one is the process of the biological development. So this all about the scope and the method of the biophysics. Now I am going to explain about the chemical foundation of the biophysics. This is the second chapter of our textbook. Now in this I am telling about the chemistry and the biology. First of all, as I told in previously, all living cells and the organisms are very complex in structure. This all exist by the chemical processes because from the outside due to the metabolisms. So by this we can say, as I told, biology and biophysics and chemistry are inseparable. They are tied up or interlinked with each other. So if we talk about like no physics method alone could not be describe the structure of the molecules. We required the chemical in that. Like chemistry only solve this problem by the studying the structure and how the reactions takes place, how it is transformed and all. And we know in previous lower classes, organic chemistry is derived from the inorganic chemistry. Because it is said to be, like scientists is said to be reliable support of vitalisms to learn how the synthesis of organic from the non-living things. Organic is made by the non-living. Now previously I talked about the biophysics. Now I am going to explain about the biochemistry. What is biochemistry? This is a science which deals with the structure and the properties of biological molecules by the interaction of the living matters. So the biochemistry mainly deals or mainly cope up with the metabolic pathway. And then biochemistry and the physics, the union of biochemistry and the physics which give up the molecular biology. We read in second year also. What is molecular biology? Which deals with the physicochemicals, molecules, interruption of biological processes. And then the organic convert into the living matter. Again it converts because of the how long it experience in the molecules. So again it converts into the living matters. Now we talk about the features of chemistry in living matters. There are 7 points in this. First point is that living system is necessarily chemically heterogeneous. They have different types of Like heterogeneous meaning is that different types of molecules. And then biological molecules takes place cannot live, that is they do not differ from any other molecules, like they do not differ. And then the second point is that living matter is characteristic by unity of chemical substance, like atom which form the element, that is a unity. The main substance and the chemical reaction are same in every living organism, take from plant or humans. Like all proteins in humans, all proteins are made up of 20 amino acids, and then nucleic acid of 4 nucleotides, and then atomic structure is same and biological process also same. The only varies in the organism is that combination of the same atomic group. This is the only reason which varies in the organisms. Third point is that the structure and the property of living cells and organism identified in the final analysis by the nucleic acid. Like after the reaction, the structure and the properties we can easily identified in the final reactions, like DNA and RNA. As we know, RNA is a synthesis of the protein, and DNA also we know that. No, and we know no chemical reaction cannot be takes further without the specific enzymes. Biochemical process and the biological molecules are resolved by the evolutionary developments. And then fifth point, there's also minor difference in the structure. Like if we take ethyl alcohol and methyl alcohol, ethyl alcohol is C2H2OH and then methyl alcohol is CH3OH. In ethyl alcohol, this causes the intoxication, and in methyl it causes the blindness. So sixth point is that chemical reaction that which takes place, it always strictly follow the forward and the reverse pathway. And the last but the not least, biological molecules are made up of the atoms, like carbon, nitrogen, oxygen, sulfur, etc. and alkali, earth metal, alkaline, like sodium, potassium, calcium, and other metals like zinc, iron, copper, etc. This all are made in the biological molecules. This is all about the features of the chemistry in the living matters and the biological development. So if I talk about in herself, like human body, it contains hydrogens, oxygen, carbon, nitrogen. They have the different percentage. The more we have is hydrogen and the oxygen, because we consume water, so that water has hydrogen and the oxygen, so we have the more level of the hydrogen and the oxygen, low level, like low percentage of the magnesium. So this is all about the chemistry and the biology. Now I'm finishing up here, and my friend Sahil will explain about the next topic, amino acid. Thank you.